I'm gonna talk about the definitions of antibiotics, the main properties, and the main classes of antibiotics in use nowadays. Okay, first, what is antibiotic? The first definition is that antibiotic is a substance produced by one microorganism that inhibits the growth of other microorganisms. However, this definition is not in use anymore because of the development of new synthetic and semi-synthetic antibiotics. So the more accurate definition of antibiotic is now the substance produced by a microorganism or by chemical synthesis, wholly or partially, that inhibits the growth of other microorganisms in low concentrations. So now antibiotic is any substance that inhibits the growth of a microorganism, but it may or may not be produced by a microorganism because we also have chemical synthesis antibiotics. Another interesting point is that antivirals and antifungals are not antibiotics, okay? Because the antibiotics only target bacteria. So antivirals and antifungals are not antibiotics because they don't target bacteria. Okay, another term that we need to know is that antibiotics have selective toxicity. What's that? Is the ability of the antibiotic to target only bacterial cells and not the host cells. So why is that? Because we need the drug to kill or to inhibit bacteria cells, not our own cells. That's the main property of the antibiotics. So how the antibiotic does that? Because they have a target that we don't have in our own cells. For example, cell wall. We don't have cell wall present in our cells. So if the antibiotics acts against cell wall, it's gonna attack only the bacteria and not the host. Another characteristic is like ribosomes. Ribosomes in bacteria are different. They have different structure in bacteria, in prokaryotic cells. So if the antibiotic acts against the ribosomes, it's not going to affect our own ribosomes, eukaryotic ribosomes, only prokaryotic cells. So this is very important to not create side effects in the host. So we are going to talk about all of them now. Natural is the antibiotics that are naturally produced by microorganisms. So we've been discovered a lot of antibiotics since the discovery of penicillin. So even fungi or the bacteria produce antibiotics. Some examples of fungi producing is Micromonospora purpurea. It produces the gentamicin antibiotics. It's an antifungal agent. And Penicillium acremonium produces Chrysophyllum, penicillin, and cephalosporin. So just remember that Penicillium species was the first species discovered to produce antibiotic that produce penicillin, and we still use nowadays. Bacteria are the most gram-positive bacteria produce antibiotics. The family Actinomycetes produce more antibiotics than any other genus. Bacillus species produce bacitracin, polymyxin, and the Streptomyces species produce 75% of the antibiotic backbone in the market. So 75% from the antibiotic that we use comes from the Streptomyces species. Why is that? Because Streptomyces, they are soil species, and in the soil they have a lot of competition. So this competition leads to more antibiotic production. So they need to compete between each other to survive. So it leads to more antibiotic production. Here is some examples of species of bacteria that produce the antibiotic that we use. These are all streptomyces bacteria. So, for example, erythromycin, streptomycin, neomycin, kanamycin, chloramphenicol, tetracycline, nystatin, and clavulanic acid, they all come from streptomyces. The synthetic is used, but is not so spread. We have some that are very useful. The most common is the sulfonamides. It was the first synthetic antibiotic created. Okay, we're gonna talk more about sulfonamides later. So synthetic is when the antibiotic is completely synthetically produced in the lab. The semi-synthetic, the difference is that they use a natural backbone to modify the antibiotic using chemical synthesis. So it's not completely synthetic, but not completely natural as well. So it's like an intermediate between synthetic and natural. So we use part of the molecule produced by a fermentation process using the appropriate microorganism. Then the product is further modified by a chemical process. Some examples are penicillins. We have the natural penicillin, but also the semi-synthetic penicillin, cephalosporin and fluoroquinolones. Now I'm gonna talk about antimicrobial spectrum. Some antibiotics have narrow spectrum. What does that mean? It means that they inhibit just a narrow range of microorganisms, usually less than 3 classes. Okay, and broad spectrum is when the antibiotic acts against more than 3 classes of microorganisms. So if an antibiotic attacks 3 or less classes, we say it has a narrow spectrum, and if it attacks more than 3 classes, we say they have a broad spectrum. I'm gonna talk about narrow spectrum first. Some examples: isoniazid, it causes inhibition of mucolic acid synthesis. It is used against tuberculosis, so it is active against Mycobacterium tuberculosis. Bacitracin, it carries off peptidoglycan building blocks, so it is active against gram-positive bacteria and Neisseria species. Polymyxin attacks bacterial LPS bind, so LPS is only present in gram-negative. That's why this antibiotic is only effective against gram-negative bacteria. And nystatin is active against these fungi, Clostridium albicans. Now I'm going to talk about some examples of broad spectrum of antibiotics. As I said, it acts against more than 3 classes of antibiotics. Okay, so these antibiotics should have reserved used because it enhances the bacterial resistance, causing superinfections. So if I use this antibiotic, my bacteria will have more chance to get resistance. So it's better to use just when it's extremely necessary. So the use should be reserved to bacterial resistant to narrow spectrum drugs. So we only should use if the bacteria is already resistant to narrow spectrum drugs, because it may cause superinfection or superinfections. Usually this is caused by overgrowth of Clostridium difficile, that is an urgent class of antibiotics. Some examples of broad spectrum are tetracyclines, sulfonamide, and semi-synthetic penicillins. Superinfections is when a second infection occurs on the top of previous infection, usually caused by the same strain of bacteria. So what's that? I got an infection and I didn't treat them properly, or my bacteria got resistant. Then I got a second infection over the first one. So it's a second infection over the first one. and this is usually caused by the same strain of bacteria. The disruptor of natural microflora may lead to proliferation of bacterial resistance, causing superinfection. So another way to get superinfection is when I get disruption of my natural microflora in my gut. Okay, so this may lead to superinfections. Now I'm gonna talk a little bit about semi-synthetic. It has some benefits because it is less toxic, like it has less toxic side effects than the original natural. It has a broader spectrum than the original, and it is more tolerant against stomach acid or enzymes produced by resistant bacteria, for example, beta-lactamase. So this improves the efficiency of the drug. So my semi-synthetic is better because it resists against my stomach acid or any enzymes that the bacteria produce, especially resistant bacteria. Because of that, it is more efficient or faster killing bacteria. Now I'm gonna talk about the classes of antibiotics. My antibiotics has different classes according to the mode of their action or the target that they have. They may have different or overlapping spectrum of activity. So, for example, two antibiotics can attack the same bacteria, so they have overlapping spectrum, or they can attack completely different bacteria, so they're gonna have different spectrum. They have different efficacy against different organisms, so each class has different efficacy, and they are subject to different modes of resistance. So depending on the mode of their action, they're gonna get different modes of resistance as well. So it may influence the activity of different classes of antibiotics. Most antibiotics must go inside the cell to kill the bacteria, most of them. Bacteria may mutate or create resistance against different classes of antibiotics. So this is a fact. Bacteria may mutate or create resistance against different classes of antibiotics. For example, bacteria who has reduced porins or increased efflux pumps make bacteria more resistant against multiple classes of antibiotics. So bacteria who has less porins, it means it has less antibiotics going into the bacteria, or more efflux pumps that pumps out the antibiotics away from the cell. They have more chances to get resistant against multiple classes of antibiotics. So we have four main classes of antibiotics. We are gonna explore each of them, but the four are: first one, antibiotics that act against the cell wall synthesis of bacteria; others against protein synthesis, so the translation; others against the nucleic acid synthesis, so the transcription; and the metabolic pathway. Usually this last one is not usually used by humans. They are more antifungal antibiotics. Okay. First, I'm gonna talk about the cell wall synthesis. So cell wall synthesis antibiotics are those that inhibit the formation of the cell wall of the bacteria. Why is that important? Okay, because if the bacteria doesn't have its cell wall properly, it's gonna explode, it's gonna die. So the first one is and the most famous is the beta-lactam. So this antibiotic, it binds irreversibly. What does that mean? That binds and does not disattach after. So they bind to the enzymes that cross-link the NAD subunits of the cell wall. So these units are necessary to build the cell wall, and For them to be linked together, they need the enzymes. So the beta-lactam binds to those enzymes, and then those enzymes are not able to do their job of linking the NAM and NAG subunits. Then the cell wall is not formed. This occurs in natural penicillins, in semi-synthetic penicillins, and in cephalosporin. We have another type of antibiotic in the same class that are the glycopeptides. They also inhibit the formation of the cell wall, but they don't bind to the enzymes, but they bind to the peptidoglycan precursors. So they bind to the NAM and NAG, so they bind to the subunits, not the enzymes. Okay, I'm gonna explain a little bit more each of them. So first the beta-lactam. So here I have my NAM and NAG, and my beta-lactam, they bind to the enzyme. Okay, so here is my enzyme. My enzyme is necessary to bind the NAM and NAG between each other to form the cell wall. So my beta-lactam binds here in my enzyme, and my enzyme is not as accessible to this subunit, so my subunit is not gonna be linked. Okay, so my beta-lactam bind irreversibly, so they don't go away to the enzymes that cross-links the NAM and NAG subunits, which are not able to cross-link, making the cell wall to disrupt. So, bacterial cells must build peptidoglycan layer in order to divide and grow. So if the bacteria does not do that, they will not be able to divide and grow. The cell wall is up to 50% of bacterial cell weight, so it's a huge part of the bacteria. If my cell wall is weak because of the antibiotic, the cell wall will not protect the bacteria against changes in osmotic pressure, and then the cell will lyse. So if I have a weakening cell wall, it will not tolerate the osmotic pressure from inside the cell, and the cell will lyse, will break down. Then the bacteria will explode, will die. Natural penicillins. I have two examples. Penicillin G. It attacks mainly gram-positive, especially Staphylococcus and Streptococcus, and it's injectable, so I need to get an injection to get this penicillin. And penicillin V is basically the same way, but can be taken orally, so we can just ingest them. There are some disadvantages about natural penicillins that are they have narrow spectrum of activity, so they are usually good for gram-positive only, because gram-negative doesn't have so thick cell wall, so it doesn't affect much, because gram-negative has another layer after the cell wall, so it's not gonna damage them very much. And another one is that they are susceptible to beta-lactamases or penicillinases that are produced by resistant bacteria. What does that do? Here I have my penicillin, my antibiotic. This is my chemical structure. What my beta-lactamase does, the beta-lactamase of the bacteria does, it breaks down the beta-lactam ring, so my antibiotic loses that function. Okay, so I have my beta-lactam here, and then the beta-lactamase of the bacteria breaks down my antibiotic. Then my antibiotic is gonna lose their function. Okay, now I'm gonna talk now about the semi-synthetic penicillins. I have these examples: oxacillin, ampicillin, amoxicillin, imipenem, and methicillin. And it has some advantages because it has broader spectrum than natural beta-lactam, and they are more resistant toward penicillinases because penicillinases are natural produced by bacteria, but here is semi-synthetic, so bacteria does not recognize this very much, and it is more readily absorbed. It requires lower doses, which reduce the toxicity. So I need to take less doses of this semi-synthetic compared with the natural. So it reduces the toxicity that this antibiotic can cause to my body. And it is also more stable in acidic environment. For example, in my stomach, it's gonna resist more to the acidity of my stomach. Now I'm gonna talk about another one. Another beta-lactam is the cephalosporin. It acts in the same way as penicillin, but they are more resistant to penicillinase or beta-lactamase. With the evolution of these antibiotics, generations become more effective against gram-negative than gram-positive. So as I said before, penicillins are more effective against gram-positive, but this cephalosporin has been evolved, and with the generation after generation, it became also effective against gram-negative. So here you can see the first generation it has a narrow spectrum. It was only effective against gram-positive. But then, from the second generation, it expanded its spectrum and became more effective against gram-negative. Now, the third generation, it became most active against gram-negative, even more than against gram-positive. And the first generation, it has the most extended spectrum of activity, so it has more it's more efficient against a wide spectrum of bacteria diversity. Okay, another type of cell wall synthesis antibiotics is the glycopeptides. The difference is that this antibiotic binds to the peptidoglycan precursors and not the enzymes. Okay, so here you can see these are the cell wall precursors, and here is my transpeptidase, the enzyme that is used to bind them. But in this case, my antibiotic, the glycopeptide, binds to the precursor, not to the enzyme. This is the main difference between them and the penicillinase. Okay, the penicillinase binds to the enzyme. This glycopeptide binds to the precursor. This is the main difference. So if it binds to the precursor, the cell wall precursor will not link and the cell wall will not be formed. This is mostly active against gram-positive again, because gram-positive has a thick cell wall, and it is also more resistant to beta-lactamase because it does not have beta-lactam in their molecule. So beta-lactamase breaks down the beta-lactam ring in the penicillin, but here this glycopeptide doesn't have the beta-lactam, so it is more resistant, doesn't get affected. An example is the vancomycin. It has a narrow spectrum, but it is a drug of less resource because it is toxic to humans, and it is mostly against gram-positive. But in some cases it's necessary to use because they are effective against methicillin-resistant Staphylococcus aureus and Enterococci. Okay, so they are effective against resistant bacteria such as Staphylococcus aureus and Enterococci. Okay, so it can act against ribosome or the tRNA, the elongation of the protein chain. I have these main types: aminoglycoside that has 4 main antibiotics. glycylcyclines, macrolides, and chloramphenicol. So I'm gonna talk about each of them now. First, aminoglycosides. They interfere with the initial steps of protein synthesis by attaching to the third S subunit of the ribosome. So it attaches to the ribosome, to the third S of the ribosome. Okay, so here you can see the mRNA. This is the third S subunit, and this is the 50, and this aminoglycoside attaches here, okay, in the third subunit of the ribosome. If it attaches here, it's gonna cause mismatch of the codon because the wrong tRNA is gonna bind here. Okay, the tRNA are able to bind, but a wrong tRNA is gonna bind here. So let's say my transcription should start here in this G, but I have the antibiotic here, so it's gonna start here. So I'm gonna cause different amino acids being brought, because if it was here, my sequence would be GCG, but now it is starting with GGA, so it's gonna bring a different amino acid. So if I have different amino acids, I'm gonna produce a different protein. So this protein may have a different function or no function at all. Okay, so this will make the bacteria to be weak. So different function or no function. So I'm gonna talk a little bit more about gentamicin. I'm still talking about aminoglycosides. So gentamicin is active against many strains of gram-positive and gram-negative, including Pseudomonas aeruginosa. It's often administered when beta-lactam It is often administered together with beta-lactam to delay the development of resistance. So the use of two or more different classes of antibiotics reduce the risk of the development of resistance. So this gentamicin is usually recommended to be used together with beta-lactam, so it will delay the development of resistance. So it's harder for the bacteria to develop resistance against the two antibiotics in the same time. Now we're gonna talk about kanamycin. It's active in low concentrations, so I don't need to get much of this antibiotic against various gram-positive, including penicillin-resistant staphylococcus, so methicillin-resistant staphylococcus aureus, and it's also good against gram-negative. This is recognized as a second-line drug to treat tuberculosis. Okay, so this is a second line to treat tuberculosis. Now I'm gonna talk about streptomycin. This was isolated in 1944 by Waxman. This was the primary drug against tuberculosis. So kanamycin is the second primary drug to treat tuberculosis, and streptomycin is the primary, okay, to treat tuberculosis. So do not get confused. Streptomycin is the primary drug, and kanamycin is the second drug to treat tuberculosis. This has high toxicity, and it leads to rapid development or resistance of bacteria. So it's a good idea to use this antibiotic in combination with other antibiotics. Okay, the last aminoglycoside is the tetracycline. It is a broad spectrum group of antibiotics produced mainly by Streptomyces. It acts against gram-positive and gram-negative. It's used as animal feed additives. So what tetracycline does? It blocks the attachment of tRNA to the third S subunit. So the previous one, both of them attach to the third subunit of the ribosome, but the first one does not inhibit the attachment of the tRNA. Okay, just cause mismatching. But in this case, this they don't allow the tRNA to bind, so no amino acid gonna be brought, no protein will be formed. Okay, so this prevents the elongation of the growing peptide chain. In the previous example, they may create a peptide chain, but it's gonna create a wrong protein. But in this case, it's gonna prevent the elongation of the peptide chain and the formation of the protein. Okay, so these all four were example of aminoglycosides. Now I'm gonna talk about glycylcyclines. They are similar to tetracycline. They also block the third S subunit of prokaryotes ribosome. It was discovered in 2000, and it is effective against multiple resistant Staphylococcus and multi-drug resistant bacterial strains. An example is the tigecycline, but it is considered a drug of last resource, okay, because it causes discoloration of teeth, it causes liver toxicity, so it may cause several liver toxicity, is very bad. So this drug may be used just as a last resource. Now I'm gonna talk about the macrolides. It's the third group of protein synthesis inhibitors. The macrolides prevent the elongation of the peptide chain by blocking the peptidyltransferase activity. So, in this case, the macrolides do not bind to the ribosome, but bind to the enzyme that puts together the amino acids. So, the tRNA is gonna bind in the correct place. It's gonna bring the correct amino acid, but if I have macrolide here, my amino acid will not be able to bind and to elongate my chain. Okay, because I need this enzyme, peptidyltransferase, and my antibiotic binds to my enzyme, and then my amino acid will not be able to bind. These macrolides are mostly effective against gram-positive and just a few gram-negative, because gram-negative are impermeable to macrolides. So gram-negative has reduced cellular uptake, so the gram-negative does not absorb much macrolides. That's why they are not effective against gram-negative. There is a natural macrolide, it's called erythromycin. It's often used as an alternative to penicillin G. It acts against Staphylococci and Streptococci. And there are some semi-synthetic macrolides. They have better tissue penetration than the natural one, erythromycin, and it has a broader spectrum of activity. Thus, it prevents the peptide chain to be formed. Okay, just like the previous one, but in this case it doesn't attach to the enzyme. It just prevents the peptide bond to elongate. It is synthesized chemically, and it's much cheaper than the isolation than if we isolate from Streptomyces. It has a broad spectrum, but it also has some disadvantages. It causes serious adverse effects, and it causes suppression of bone and neural activity, and should not be prescribed if there is an alternative. So, because it causes serious adverse effects, it should just be prescribed if there is no alternative. Okay, now I'm gonna talk to the third group of antibiotics. Is the antibiotics who inhibit the nucleic acid synthesis. So the first one is the rifamycin family. This rifamycin family inhibits the mRNA synthesis. It's often used against mycobacteria in the treatment of tuberculosis and leprosy. It's able to penetrate tissues and to reach therapeutic levels in cerebrospinal fluid. This is a valuable characteristic, okay, because this is very important. But resistance develops very quickly because bacteria develop mutation in RNA polymerase. Okay, so I need RNA polymerase to form my mRNA. But this antibiotic inhibits the mRNA. But if the bacteria develop mutation in its RNA polymerase, my antibiotic will not be able to inhibit the mRNA synthesis anymore. Another example is the fluoroquinolones. It inhibits the DNA synthesis by binding to DNA gyrase that are topoisomerase 2 and 4. So first I'm gonna explain briefly what's DNA gyrase and topoisomerase. So they are enzymes that promotes the unwield of the double strand of DNA when the DNA is being duplicated. If I don't have this enzyme here, my DNA will be tossed and my duplication will stop, or even my double chain of DNA will be broken. Okay, so these are enzymes that participate in the overwind of DNA during replication. This is very important. If these enzymes does not act, the torsion will enable the DNA to replicate, or even will damify the DNA, will break down the DNA. Okay, so now what fluoroquinolones does? They bind exactly to the topoisomerase. So here is my topoisomerase enzyme. My fluoroquinolones bind exactly in this enzyme. So if my fluoroquinolones is here, the topoisomerase will not make their function and the duplication of DNA will stop, or even the DNA of the bacteria will break. Then so bacterial DNA synthesis is inhibited. It has a broad spectrum against gram-positive and also gram-negative. And the major representatives are nalidixaste, is just quinolone, norfloxacin is fluoroquinolone, and ciprofloxacin is also fluoroquinolone. Fluoroquinolones have much wider spectrum of activity than traditional just quinolones. This is the antibiotic of choice against urinary tract infection and pneumonia because it's not toxic. Okay, so this antibiotic is not toxic. But there are disadvantages is because bacterial resistance can develop very quickly. Okay, the last group now is the inhibition of metabolic pathway. The most famous is the sulfonamides. This antibiotic targets the metabolites, not enzymes, that are essential to synthesis of DNA and RNA nucleotides. So here, for example, I have this metabolic pathway, and this is when I take some components to create my DNA and RNA. So this is an anabolic process because I take several small subunits or small components to create a more complex molecule. Okay, so this is an anabolic process. Okay, so these antibiotics, they act exactly to the enzymes that make this possible. Okay, so sulfonamides, for example, act in this process. You don't need to know exactly where, you just need to remember that it affects the metabolic pathway to form DNA and RNA, and this is a anabolic process, so it acts against a anabolic process. Sorry, my letter here. Sulfonamides, they were the first synthetic antibiotics to be produced, and this means that they are completely produced in the lab. They have a broad spectrum, and they are used to treat urinary tract infection. They are bacteriostatic. It means that they stops the growth of bacteria, but not necessarily they kill all the bacteria that are present. They just stop the bacteria to grow that population. But the development of resistance is very common in this antibiotic. Okay, so thank you so much. I hope this helps.